Right, good morning, everybody, and welcome to Adirondack Lake Ecology and How Invasive Species Disrupt Aquatic Ecosystems. My name is Sean Kittle, and I am the Communications Coordinator with the Adirondack Park Invasive Plant Program, and I'm going to get us started this morning. And just a heads up, this webinar, like all of our webinars, is being recorded and it will be uploaded to our YouTube channel, usually within about 24 hours. And if you've registered for this, that's great news because I will send out an email with a link and some other information following the webinar. So today's speakers are Peter Tobiessen. He's the professor emeritus at Union College and author of The Secret Life of a Lake, The Ecology of Northern Lakes and Their Stewardship. He's going to cover the first portion of our talk today, and then he'll hand it over to Brian Greene, who is the Aquatic Invasive Species Coordinator with the Adirondack Park Invasive Plant Program, also known as APIPP. So today's trail map's pretty straightforward. I'm going to do a quick welcome here, get into a little bit about what APIPP is and what we do, just for those of you who don't know. Then I'm going to hand it over to Peter, who's going to talk about Adirondack Lake ecology, and then we'll have a Q&A with Peter after that. After that, Brian is going to talk about how invasive species disrupt aquatic ecosystems. He's also going to talk about the APIPP Lake Protectors Program. It's a volunteer program. And he is going to close out with a message on how to prevent the spread of aquatic invasive species. After that, we'll have a final round of questions. And for the questions, we ask that you please type your questions into the chat. You can do that while the speaker is speaking. We just don't like to have the speakers get interrupted. Then once we get to that Q&A time, we will go through that chat and make sure all the questions are answered. So the Adirondack Park Invasive Plant Program is one of 8 partnerships for regional invasive species management across New York State. We call those PRISMs, P-R-I-S-M for short. And our service area is the Adirondack Park and north up to the Canadian border. Our program is funded by the Environmental Protection Fund, which is administered by the New York State DEC. And we are housed in the Nature Conservancy's Keene Valley office, which of course is right here in the beautiful high peaks of the Adirondacks. APIPP's mission is to work in partnership to minimize the impact of invasive species on the Adirondack region's communities, lands, and waters. Since invasive species are primarily spread by human activity, our outreach efforts largely revolve around public education and prevention. Basically, it's a lot easier to prevent invasive species from spreading than it is to deal with them once they're here. We also empower the public through trainings where we teach people to identify invasive species and to do their own management of any invasives that might be on their property. We work out in the field, and our team focuses on managing priority infestations, and we also work on land and in water. We have partnerships with about 30 organizations around the region, and those partnerships really help us amplify our efforts, reach more people, and cover more ground, as do our volunteers. We have volunteers who have been with us for more than 20 years. It's really impressive. And they're out surveying lakes and trails for invasive species and reporting any invasive species that they find. So again, Brian's going to get more into how you can get involved by volunteering for our Lake Protectors Program. And we also have a terrestrial version of that called the Forest Pest Hunters Program that you can also get involved with. You can learn more about us at adkinvasives.com. So please check out our website and learn more. And from there, I will hand it over to Peter. Thank you.
Thanks, Sean.
You're
welcome, everybody. I'm going to talk about the Adirondack Lakes, and we'll get started here. Whoops, I just pushed the button and the slide didn't change.
I'll make sure you request access to my screen, Peter.
You can control the screen. Okay, is that it? Use my keyboard. Is that what I say?
Yeah.
Well, it's not doing it. View options.
If you'd like, I can advance the slides if you just say next.
Use Sean Kittle's keyboard layout. I did hit use my keyboard layout. Is that what I'm supposed to click on?
Why don't you just say next and I'll advance the screen for you.
Okay, great. Okay, good. Let's see, I'll get out of here. Okay. Okay, next slide. Here is a typical food web of a lake, and it's essentially the outline of what my talk is going to be about. First, I'm going to talk about water, and then the nutrients, and then the organisms, starting with the algae, and then up into the smaller animals, and then the bigger animals. So this essentially is an outline of what my talk is all about. Okay, next slide. Water is weird. Water it behaves differently from all other liquids that you are familiar with. Typically, a liquid will increase its density, become heavier, as temperature decreases until it becomes a solid, which weighs much more than its liquid form. Water is different. As you can see from this table, as water in a lake cools down during the fall, its density increases. So as it cools down, its density in grams per centimeter cubed increases until 4 degrees centigrade or 39 degrees Fahrenheit. This is water at its most dense. As a matter of fact, this is the definition of what a gram is, weight of water in a cubic centimeter. From here on in, the temperature of the water can decrease, but keep going down, but the density decreases too. So the water here is lighter than the water here. And an ice is much, much, much less dense. So it's much lighter than water at any temperature. And this is why ice floats. In the winter, you'll have ice at the surface and liquid water all the way down to the bottom of the lake so that we can maintain biological communities even in the winter. If water behaved like other liquids, the ice would be heavier than the liquid water. As the lake cooled down, the ice would fall down to the bottom of the lake and begin building up. And if you saw ice on the top of the lake, the entire lake would be solid ice. And so we should all, as lake lovers, be happy that water is weird. Next slide. Next slide. This is an experiment that I do in my aquatic biology course to illustrate what happens in a lake. So this is my micro lake. It's an aquarium that's about 1 foot high and 1.5 feet long and about 4 inches deep. A couple big floodlights up here to simulate the sun, a fan over here to simulate the wind, and I fill the lake, this aquarium up with cold water, and I throw a lot of ice in there, and I bring it cold enough so that the ice floats on the top and it doesn't melt. And then every 10 minutes, students measure the temperature profile about 2 centimeters apart, down all the way down to the bottom, and we then record the data. So next slide. This is the data from this experiment, which is right in front of the class during the class period. So we see the ice the water is very cold here, and then this is the temperature of the water going down until it reaches 29 or 39 degrees centigrade, and then it's the same all the way down to the bottom. Now we turn the lights on, and of course the lights produce a lot of infrared radiation, and water is very good at absorbing infrared radiation. So the top of the water gets warm, and after 10, 20, and 30 minutes, it gets very warm, and there's a very, very sharp gradient in temperature from the top to the bottom. And then if you turn the wind on, it mixes up the upper layers a little bit, but it's still, notice that the bottom of the lake, my little micro lake, is still cold. So next slide. This is what we find in every lake that you look at in the summer that's deeper than about 20 feet. We have an upper layer in the water that is warm, called the epilimnion. And then there's a gradient, steep gradient down to the lower level, which again is fairly stable. The temperature is pretty stable, called the hypolimnion. And in between, this is the thermocline, which is the gradient in temperature. Next slide. Now, the most important thing about this experiment is that at the end we leave the lights on and the fan on, and you put a little drop of dye, blue dye, in the top layer. And it spreads, since the fan is there, there's turbulence here, it spreads throughout, but it does not go into the bottom of the lake. This gradient, this temperature gradient called the thermocline, isolates the top from the bottom. So the top water does not mix with the bottom water. And this is very, very important when to understand in lake ecology. When you look out on a lake in the summer, you're really looking at two lakes, a warm lake on the top and a cold lake on the bottom. And they are very different, not only thermally, but also chemically, and quite different as far as the biological communities that are in them. Next slide. So this is what it looks like. This is a profiles in a real lake. This is Sacandaga Lake, which is a small lake near Speculator. It's not the reservoir. We're very snooty about that. So this is a natural lake, and in the winter, it's this typical cycle. We have this ice on the top. These are temperatures in centigrade now, and this is depth. Ice at the top, and then these temperature going down to going up, I'm sorry, to the 39 degree and 4 degrees centigrade, all the way down to the bottom of the lake. So now, as the ice melts in the spring, the temperature becomes isothermic here, and any wind that goes through stirs it up. So the water can be stirred up, so it warms up to about, in the Sacandaga, about 11 degrees, and right about then the thermocline begins to form. And when the thermocline forms, the bottom layer is not affected by what's going on the top, so it stays the same temperature, 11 degrees, all season long. So in June, we have the warm upper layer here and the thermocline. In August, it gets a little bit deeper, a little steeper. September, a little deeper and steeper. And then as the fall goes along, the water begins to cool, and the top of the water cools down until the thermocline breaks, and the entire lake is the same temperature. And now it can mix again. So the top water mixes with the bottom water in this period. And this period is called the turnover. And we have spring turnovers that I talked before, and the fall turnovers. And that's when both layers of the lakes mix up. And of course, eventually in the winter, we get the same winter profile. Next slide. Now this just compares the fake lake, the one that's on my desk in the beginning of the lecture hall, and this is the real lake, and you can see the similarities in the next slide. Whoops. Okay, as the season progresses, the upper layer has high light intensity, and atmospheric oxygen diffuses into the water, and the algae produce oxygen through photosynthesis. And this is the equation for photosynthesis, and these are the reactants, and these are the products. So oxygen is produced when the algae photosynthesize. So the top of the lake has dissolved oxygen, almost saturated all season long. So the dissolved oxygen remains high in the top layer of the lake. In the lower layer, there's less light, so there's less photosynthesis. The thermocline prevents atmospheric gases from entering it, and the sediment and organisms are dying, and when they decompose, they use oxygen in their respiratory process. So here oxygen is being used during respiration, and all of these things tend to reduce the dissolved oxygen levels as the season progresses. Next slide. So this is how the thermocline relates to dissolved oxygen. Here's the temperature with the thermocline here, and here's the dissolved oxygen. So again, the top and the bottom of the lake are very, very different with regard to dissolved oxygens toward the end of the summer. If it gets low enough down here, then the fish can't survive back there. And this is why a lot of times fishermen are fishing right around that thermocline, because that's where the fish tend to be. Okay, next slide. So this is a real from Sacandaga. So when ice is out, the oxygen level is really quite high because gases are more soluble in water under cold temperatures, and this is May, right after ice off. In June, we have thermocline here, and notice that already in Sacandaga, which is a relatively good, good water quality lake, the lower level is being depleted of oxygen. And by August, it's depleted more. And by September, it's completed a lot. So that much oxygen is lost in our bottom layer, the hypolimnion of our lake. And the implications of the level of oxygen down here have chemical results, and we won't get into them, but let's go to the next slide. Okay, now when we think about a lake, whether it's a nice lake or not, we often think about the water clarity. We have this bias toward clear water lakes, and the Adirondacks has lots and lots of them. So water clarity is a critical function or factor in evaluating lake quality, and we measure it with a Secchi disc. This is a Secchi disc. It's about 8 inches in diameter, and it's got quadrants, alternate black and white quadrants. It has a calibrated line to it. So we tie that to the disc, dump the disc over the edge of the boat, and see how far down it goes before it disappears. Okay, and we record that number. And then we lower it a little bit more, and we bring it back up and see how deep it is that it reappears. And we take the average of those two numbers, and that is the Secchi disc number. The Secchi disc depth is affected by many things, but most important is the amount of algal growth in the lake. Next slide. So here are some Secchi depths of various lakes that you're familiar with. Crater Lake, 132 feet it goes down before that disc disappears. Lake Tahoe, which is beginning to have more difficulty with water clarity, at least back when this was measured, was 100 feet. And Lake George, 27, Champlain less, Sacandaga Lake, 17, it's about 5 meters, and a nutrient-rich lake in Scotia is about 3 feet. And I was doing an ecological impact statement on a lake in Albany, and the Secchi disappeared in 4 inches. It was essentially raw sewage flowing into that lake. Next slide. So what is actually making the algae grow in a lake? So this experiment looks at the limiting factor, the nutrient limiting factor to algal growth. And I made a wooden frame that had floats on each end, and then it has 12 compartments to it. And in each compartment, I've installed a clear plastic garbage bag that I fill up with 10 gallons of water. And I anchor it out in the middle of the lake and put nutrients in each one of these different nutrients in each one of these compartments, and we leave it there for a week. So this is the next slide, please. So this is the scientific methodology here. So these are controls where we put no extra nutrients in. Here we put nitrogen in, phosphorus in, nitrogen and phosphorus, and then there are 4 replicates of that experiment. And we leave it out in the lake for a week, and we hope a motorboat doesn't run it down. Then a week later, we come back and we collect water from each of these, and we measure the amount of chlorophyll, which indicates how much algae is in there. Next slide. So these are the data. Whoops, spacing is weird here. Anyway, in the control, it comes about the 6 parts per billion of chlorophyll. So nitrogen, the additional nitrogen, it's still about 6. So there's really no difference between those two numbers. So nitrogen does not affect the growth of algae in our lake. If you put phosphorus in, you can see it more than doubles. And nitrogen and phosphorus, you get a little increase, but those numbers are not significantly different. So the result is that phosphorus limits algal growth in Sacandaga Lake and essentially all other lakes measured in the Adirondacks and elsewhere. Next slide. So phosphorus is relatively rare in most lakes. It's down into the parts per billion range. So how much is a part per billion? Consider Olympic-sized swimming pool. It's 50 meters long, 25 meters wide, and 2 meters deep. And so therefore it has 2,500 meters cubed. So this is all calculations to figure out how many actual kilograms of water in there. And it comes out to 2.5 million kilograms of water. So if we want to put 1 part per million of something in that lake, we should put in 2.5 kilograms. Okay, so if we put 2.5 kilograms of sugar into the pool, that will equal 1 part per million. And a kilogram is about 2.2 pounds, so that 2.5 kilograms is somewhere between 5 and 6 pounds. So that's essentially a 5-pound bag of sugar into a Olympic-sized swimming pool. That's 1 part per million. And 1 part per billion is 1,000 times less, or 2.5 grams. Now, how much is that? The weight of a nickel is 5 grams. So you cut a nickel in half and you put that much phosphorus in an Olympic-sized swimming pool. That's 1 part per billion. So we're measuring things at pretty small levels. So just adding a little bit more phosphorus into the lake through human activity can have a major effect on the lake. Next slide. So where are the sources of phosphorus? Wastewater outfalls in large lakes, septic leakage in smaller lakes. The Adirondacks, many of the summer, especially the summer camps, have septic systems. And the Adirondack soils are not very good. They're sandy. They're not very good at binding phosphorus. So that's also a source. And of course, they have to be maintained too. Runoff from impermeable sources like roofs and parking lots and roads. Decomposition of organic material, but in terrestrial areas, the phosphorus is readily taken up by the deep-rooted plants, so not much of that gets into the lake. Internal loading from lake sediment. The lake sediment is a tremendously rich source of phosphorus, and as long as there's oxygen down there, that phosphorus is tightly bound to the sediment. But if the bottom layer of the lake loses its oxygen level, becomes anoxic, then that phosphorus can be released into the lake. Lawns, we really haven't thought much about lawns, but lawns that come to the shoreline leach about 100 times more nutrients into the lake than a forest. So we should all consider native plant vegetation buffers between the lake and the lawn. So next slide shows a schematic. Whoops. Oh, okay. So this is some quantitative information about the amount of phosphorus that leaches into a lake from lawns and from forests. And the blue thing is groundwater, and the green is surface runoff. And look at the difference of surface runoff in a lawn relative to the woods, a forest. So 170 acres of forest is equivalent to 1 acre of lawn as far as nutrient input to the lake. So vegetation buffers should be considered. Next slide. This is sort of a schematic of a lawn. And how many of these do you see in our lakes? So many people want to have this beautiful lawn, and it goes right down to the lake. And why they want to come to the Adirondacks so they can mow their lawn, I have no clue. But another possibility is to leave many more of the trees and shrubs around and have this vegetative buffer between the lawns and the lake. And these vegetative buffers don't have to be very big. Next slide. Next slide. Oh, good. Okay. So here we have some deep-rooted plants, which is the vegetative buffer. And notice that the view to the lake is not altered. Next slide shows that these buffers can actually be very beautiful if you want to do it. And another advantage of these buffers is that they keep Canada geese off of your lawn. and that can be significant. Next slide. Okay, now we should be monitoring our lakes as well as much as we can, because lakes can go bad very quickly. So here we have a lake which has been about 20 to 25 parts per billion phosphorus for many, many years, and all of a sudden things went south and we get a tremendous increase. This lake is considering putting sewage treatment sewage pipes all around would go in. Anyway, get rid of their septic systems and use a municipal sewage system, which gets expensive, of course. Next slide. Now, EPA had studied these lakes, and this graph shows the number of cyanobacteria. Now, cyanobacteria is a fancy name for blue-green algae, which I prefer because it's more descriptive of what we're talking about. This is a group of algae which is very primitive. It's been around for about 3.5 billion years, and it is responsible for all of the oxygen that's in our atmosphere. So they're pretty nice guys, except that they can have some problems too. So here we have phosphorus, and notice that there's this inflection point between about 20 parts per billion and 30 parts per billion of phosphorus. And this inflection point gives a lot more bacteria, cyanobacteria or blue-green algae, and this is the probability that you have the lake 50% more of your algae are cyanobacteria. So this inflection point is an important thing to remember. So remember, 20 to 30 parts per billion. Okay, next slide. Now, why are you worried about blue-green algae? These are also called HABs, which is a harmful algal blooms, mainly because they can produce toxins. And 3 of the species that can do that are Microcystis, Anabena, and Aphanizomenon. I love that word, Aphanizomenon. And who are the toxins? Microcystin is one of them, from Microcystis here. It's a liver toxin, can produce digestive issues. Anatoxin, the common name of anatoxin is the very fast death factor. That should give you a clue that maybe we don't want it in our lakes. It's a neurotoxin that can cause respiratory paralysis. In some farm ponds that are very, very green with blue-green algae, there have been cattle which have died because they've drunk its water. So this is a nasty enough thing to kill a cow. And there's some evidence that they might be killing elephants in Africa. Next slide. Now, all lakes contain blue-green algae, even lakes like Sacandaga with phosphorus levels below 10 parts per billion can have small transient shoreline blooms. And in the end of the summer, when the water is warm, the water is still, there's no wave action. Algae, the blue-green algae can float to the top of the surface. They have gas vacuoles which fill up with oxygen, and they can float to the top of the surface. And it looks like an oil spill. But if there's a little bit of wave action, these things will dissipate really quickly and they'll just be gone. They totally disappear. So they're not really a big deal, but they can form, and you should be vigilant to see if they're on your lake. Next slide. Now let's look at Lake Erie. We're all familiar with Lake Erie. It has 3 basins: the East Basin with Buffalo in it, the Central Basin with Cincinnati, and the West Basin with Toledo in it. And these are the typical phosphorus levels there. So in the East and Central, there are from 17 to 6. But in the West Basin, they're higher, and they're in the magical 20 to 30 range. So what happens there? Next slide. Algal blooms on Lake Erie add toxins to Ohio's drinking water. Toledo had to change where it got its water from. It used to get it, or usually gets it, from the lake, but with these algal blooms, they had to find an alternate source of their drinking water. Next slide. So this is what that bloom looked like in 2013. Here we have Buffalo. This is the eastern basin. This is the Central Basin, and this is the Western Basin. And this is what it looks like from space. little bit closer to the water, but you see a boat going through it. This is the boat's wake. I mean, this is really disgusting stuff. And when you get in your personal watercraft, it looks like you're moving through pea soup. During these blooms, several dogs often die, and maybe 100+ people are made sick of intestinal issues from the association with these waters, with these blue-green algae blooms. Next slide. So now we're going to look at what other creatures are in these lakes. Remember the next slide. The food web here. So here we have the food web. We're going to look, we looked at the water, we looked at the nutrients, we looked at the effect of nutrients on the algae. So we'll look at the algae and then the smaller animals and then the bigger animals. And of course, the big animals eat the smaller animals and the smaller animals eat the algae. So next slide. The first group we'll look at are the phytoplankton, and phyto means plant, and plankton means small critter that doesn't swim very much, so it's wafted around in the current. They're often very, very microscopic. Now, we measure microscopic things in the unit of a micron. A micron is 1/1000th of a millimeter, and a millimeter is 1/1000th of a meter. So a millimeter is the same size as the thickness of a dime. So think of a dime and think of how thick that is, and we'll be doing comparisons with that. So if we find an alga which is 10 microns long, that means you have to pile up 100 of them nose to nose to make the size of the thickness of a dime. So these critters are really small, and you need a good microscope to see them. The phytoplankton are plants, so they are photosynthetic and they add oxygen to the water, but of course you need light for that. They're the basis of the food chain in open water and very important to water quality, and quite diverse in shape and function. Next slide. So this is a typical group of algae from a lake just outside of Schenectady in Scotia. And of course, it's concentrated, but look at the variety of all those critters in there. These are all plants, these are all algae, and they really are quite astonishing when you look at them under the microscope. Next slide. The algae are grouped up into groups relative to their pigments. We already talked about the blue-green algae. These are the green algae, and obviously they're nice and green. So here are a couple of them, and next slide shows another one. Aren't they cool? Now, next slide. Now, this is a filamentous algae. It's about 10 microns across here. Actually, maybe 100 microns across there. So it's obviously it's obvious to see, but the chloroplast is spiral. And the scientific name for it is Spirogyra, for obvious reasons. Now, not so obvious is why a British rock band calls itself Spirogyra. And I've always wondered whether their bass player studied algae, I suppose. I don't know. Next slide. Now, another group is the diatoms. These are scanning electron micrographs of them, and they're about 10 microns in diameter. So what does that mean? That means you have to pile up 100 of them end to end so that they come to the width of a dime. So they're quite varied. They have a silicaceous cell wall, which does not let oxygen or gases through. So they've evolved whole pores, and the pores are all different, and the shapes are all different. So this is just a few of the magnificent group of diatoms, and we'll see them in our lake later on. Now, next slide shows at the end of the 1900s, people decided that these are so pretty that we should make these little arrays of them in a nice symmetrical way. So all of these are different species of diatoms. This one is about 10 microns long. So this entire array is smaller than a period, a printed period. So you obviously need a microscope to see them. And actually, then can you imagine assembling all those? They're just really remarkable. Next slide. And some people have way too much time on their hands. Next slide. So this is the community, the algal community in Sacandaga Lake, which is a pretty nice lake. And you can see that there's a nice variety of algae. The blue-greens, there are a little bit of blue-greens in there, mainly green algae. Diatoms are periodic. They tend to come in the spring and the fall, and then there are other groups of algae. So this is a nice, diverse group of open water algae. Next slide. This is a not so nice group. So this is an algal composition of a lake that is dominated by blue-green algae. So again, here is the algal community, and look at how many, the domination of the blue-greens in that community. Now this lake is beginning to form toxins. So microcystins are beginning to show up. You can see them here and here and here, and even a little bit of anatoxin, the fast death factor, is present in one of the samples. So this lake has some significant issues with it. Next slide. So now we looked at the algae. Now we're going to look at the unicellular critters, the protozoans, and then into the rotifers, and then into the water flea, and then we'll go a little bit higher into the food web. Next slide. So these are the protozoans. They're single cell animals. This is a paramecium, which some of you may have learned about in high school biology class. They're very common, and they're filter feeders. So this is one cell. These are all one cells. Now this is Giardia. This is the critter that gives you beaver fever. And it's a one cell critter with a flagellum. And these are two other typical ones. So they're very small. They're filter feeders. They're eating bacteria and tiny algae. Next slide. This is a little bit bigger group, the rotifers. That can be 100, they can be 1,000 cells or so. And they have some flagellae here, which wave around and bring water currents in, which then bring microscopic critters like bacteria and small algae in where it eats it. Next slide. Here's another one. You can see it's been eating algae and has a little eye spot there. They can sense light, but they obviously can't focus that on anything. So this one is about 100, maybe 130 microns long. Next slide. Now this one's really cool. This has these little paddles on the side. There are 12 of them. And if it senses turbulence in the water, which is what it would feel if right before it gets eaten by a filter feeding critter, it flips these little feather-like things. There are a dozen of them, and it can jump 15 body lengths in 5/100ths of a second using these 12 paddles. And this is the way that it avoids predation. Next slide. Here's another one. You can see the flagellae here and the eye spot. And this one has a little leg coming down with the 2 fingers there, so it can grasp hold of something, and so it can be stabilized in the community. Next slide. So this shows where rotifers are in Sacandaga Lake, and it shows the importance of the thermocline, and it shows also that the distribution of critters is determined by the kind of water that it's in. So here is the thermocline, and here's Polyarthur, the guy with the little paddles, and you can see that he is mainly in the upper warm water of the lake, not so much in the bottom. This guy here, Calicatia, is only in the bottom of the lake, the bottom cold part of the lake. So these two guys are not competing with each other. This little guy here is not as common, but more common actually in the thermocline. And the one here, Trichoceras, is only in the upper level and not down in the bottom layer. So it shows the segregation of the organisms in the lake relative to the thermoclines. It's easy to look out at a lake and think that it's a totally homogeneous place, but it is absolutely not. So you can see the segregation of the populations of rotivores. Next slide. Now this is what a water flea. And they have these great big appendages in their front that they use like oars, and they beat them back, and so that this little critter goes bouncy. He jumps back and jumps along. And the first person who named them said, "Boy, that looks like a flea because fleas jump. So let's call this a water flea." Well, it's not an insect, but the name has stuck. This is a filter feeder. And these are his filtering appendages here, and here's a blow up of that. And they wave these through the water, and they gather up small critters along here that won't go through that get stuck in their filter. And then they work that food up, put it into a ball, and with these appendages. And here is the gut. And here they have a brood patch where she raises her little babies, and they have a cute little eye. They're really sort of neat critters. Next slide. Here are some in real life. You can see this guy's been eating algae, his eye and his appendages, and this is a top view of them, and you can see their appendages and their brood patch. And I like this guy because he looks like an elephant. Again, he's been eating algae, and those are her babies there. This is one genus of them that is much, much larger. So this is the thickness of one dime here. So this thing is probably 2 or 3 centimeters, not centimeters, millimeters long, and it's obviously very obvious to see, but with your naked eye. These are actually about 2 millimeters, so you can see these with your naked eye. So it's not like the algae and some of the smaller critters. Next slide. There's an invasive spiny, a water flea in there called the spiny water flea for obvious reasons. That's its spine, and it clogs up fishermen who are trolling, and their lines are down in the deep water. These can gather up they get tangled up with their long tail, and they can just make a big wad of water fleas on the fisherman's line. Also, they've been thought to decrease the other water fleas to compete with them, compete with them, and that's not a good idea. Because obviously the native critters are much more beneficial to the lake than these guys are. Next slide. So here we go. We've looked at algae. We've looked at these guys. We looked at the water flea, and now we're going to look at this group, which is a group of insects. So next slide. Here are insects that have a one stage of their life cycle, which is in the water. And they can be in the water for a year, and some of them for maybe 2 years, and then they metamorphose into adults where they're in the air, and they can fly around and mate and lay their eggs. So these are the aquatic stages, these are the terrestrial stages, and some of them, in the case of the mayfly, can be an adult for only a few hours, and some of them for maybe just 1 hour. So they don't live very long in this stage here. And as a matter of fact, the scientific name for this group is the Ephemeroptera. Next slide. Here's the mayfly. Here's the aquatic. Notice it's 2 tails. It's had gills here, and it metamorphoses into these adults with the big sail-like wing, and this nymph will swim to the top of the surface of the water and metamorphose into one of these adults. And then the adult sits on top of the water and for a few minutes or so before its wings expand and before it flies away. And trout love these guys. And so that when you're in the evening and it's nice and calm and you look out on the lake and you see these little circles of water, That's the fish that are eating these things right off the surface. If you go out and canoe among them, you'll actually see these guys come up to the surface and fly away. And they can come up and make a hatch where they all metamorphose at the same time. And that's so you can get hundreds, if not thousands of them in some cases, and they can become quite a nuisance. Next slide. Here are stoneflies. Notice he only has 2 tails. The mayfly has 3. And this is the aquatic stage. This is the adult stage with its wings flat. Now they metamorphose out in a terrestrial situation on rocks and logs and things like that. So you can see their exoskeletons on these logs and rocks near the water, and then they fly away and mate and lay their eggs. The next group, next slide, are the caddisflies, and they have they look like a grub. They said so they don't have much armor in there. So what they do is they create their own little house, and they make their house out of sand or pine needles or leaves or something like that. And here is a rock that has a lot of these little houses where the caddisfly has actually flown away. And this is the adult, and it has wings that sort of look like a little tent. Okay, next slide. Somebody decided that let's bring these into the lab, and instead of giving them sand or spruce needles or something to build their house, let's give them a little bit of turquoise and maybe a pearl or 2 and some bits of gold. And the caddisfly uses them to make its little house. So here you have a whole bunch of caddisflies with their beautiful jewelry houses. And then when they leave the house and metamorphose and fly away, then the house remains, and you can make jewelry out of it. So you can give each other caddisfly jewelry, and you'll be the only person on your block to have jewelry made by a bug. Next slide. Here we go. So we've looked at these guys. Now we're going to get into the fish. Next slide. So the fish, we have the smaller fish, the alewives. Remember that. We'll get back to the alewives. Sunfish, minnows, and everything. They eat zooplankton. So they're small and they eat zooplankton. So they're called planktivorous fish. So these are the planktivorous fish. These are the fish that eat other fish. And we call these piscivorous fish because Pisces is, I forget whether it's Greek or Latin for fish. So this is a piscivorous fish, a northern pike, and or maybe a pickerel, I suppose. Anyway, and the alewives, it's only about 6 inches long. Next slide. And then there are the plants, and Brian's going to talk about invasive plants. So here are 3 of them. Here is the curly leaf pondweed and the milfoil, which he's going to tell you more about, and hydrilla. They're limited by light intensity, so they tend to grow in shallower parts of your lake. The clearer the water, the deeper they can grow, and some can grow in water that's 20, 25 feet deep in very clear lakes. The relationship between plants and algae. If there's lots and lots of algae in your lake, the water is cloudy and it doesn't penetrate very deeply. So you don't get as many of these vascular plants. However, we've had situations like Saratoga Lake, which used to be very cloudy with algal blooms. They cleaned it up, and then the plants just expanded dramatically. And there's just an amazing growth of vascular plants in Saratoga Lake right now. So some fish eat the plants directly, like grass carp, but most of them benefit because of the habitat that they form. Fish can hide in there. They have their nurseries in among these plants, and these plants have lots of critters growing on them. And the smaller fish and the minnows and things can eat those small critters that are off of the leaves and things of the plants. There's also a habitat for wait fish predators, sit and wait fish predators. And many of these predators, like bass and pickerel and things like that, have stripes on their body, which gives them some camouflage in among the weed beds. Next slide. Okay, now suppose your lake turns into a pea soup mess. How would you want to change that? So here you have the algae, and this is what makes it a pea soup mess. So the nutrients come up here. Maybe if you limited the nutrients, you could clean up the lake. So next slide. This is an example of Lake Washington near Seattle. Here is Lake Washington. Here's the city of Seattle, and here's the Puget Sound. All of the communities around Lake Washington had wastewater treatment plants, and they dumped the effluent right into Lake Washington. And it got pretty skanky. The Secchi disk was 3 feet, chlorophyll 30 parts per billion, phosphorus 65. Remember, 20 to 30 was the inflection point, 65 parts per billion, and blue-green algae 90%. So what do you do? They tested the water, found that the limiting factor was phosphorus. They also noted that lots of phosphorus was coming out of these wastewater treatment plants. So they started a project to divert the wastewater outfalls into Puget Sound. Now, this is a huge sound with tidal waters. And in fact, the solution to pollution is dilution, but in fact, it didn't have create much of a problem in Puget Sound. And it was completed in 1968. So next slide. Oh, my word. Wow. So this is before the project, 3 feet, 31 parts per billion, 60 and 90%. After the diversion of the wastewater, the Secchi went from 3 feet to 23 feet, from 31 parts per billion to 3 parts per billion, 65 to 17, and from 90% to 10%. So this was a successful thing, and the reason they could do it is that the source of the phosphorus was a point source. They knew it was coming from the wastewater treatment plants, so they could control there. This is considered a bottom-up solution because on the food chain, it was coming from the bottom of the food chain up to the algae. Next slide. Okay, in Lake Mendota in Wisconsin, they had an algal problem, lots of blue-green algae, poor water quality. Phosphorus is the limiting factor, but the sources were diffuse. Lawns and agriculture and urban suburban land use over the entire watershed. So a bottom-up control strategy would be difficult to impossible. Is there another solution? Next slide. Again, we go back to the food web. So the bottom-up will not work. So let's see what we can manipulate with the food chain in order to get to eat these algae. So next slide. So they proposed a solution. Who eats the algae? Zooplankton. Who eats the zooplankton? Planktivorous fish. Who eats the planktivorous fish? Piscivorous fish. So they stocked a lot of walleyes and northern pike to eat the planktivores. So if you eat the planktivores, then they don't eat the zooplankton, and the zooplankton flourish, and they will eat the algae. Okay, so they stocked with piscivorous fish, but who eats the piscivorous fish? Next slide. Ah, yes. Next slide. I don't eat my own fish, so that was a joke. But the wily Wisconsin anglers do, and fish pressure on Lake Mendota went from 180,000 person hours to 460,000 person hours in 2 years. The walleye harvest went from 22% to almost 60%, which was unsustainable for the hatcheries. The conclusion was the top-down trophic manipulation won't work if you can't control the top predators. But it has worked on smaller lakes with controlled access. Next slide. Like Otsego Lake. It had decreasing water quality. It had No point source of phosphorus, no evidence of recent development or anything like that. Research showed that the culprit was an invasive fish species, the alewife. Remember the alewives we looked at in about 20 slides before? 6-inch fish, which eats a lot of zooplankton. The reduced zooplankton community allowed algae to flourish. Next slide. So they brought in big fish just like they did in Wisconsin. So the zooplankton, in just a few years, and the alewife population crashed. Zooplankton populations increased and algal populations, blooms, decreased, and the water quality increased, the water clarity increased. For some reason, the New York anglers didn't increase fishing pressure like the Wisconsin guys, and wonder why. Maybe less public access. Maybe they didn't know about it. Maybe they're not as good at fishing as their Wisconsin peers, or maybe New Yorkers just don't like walleyes. I don't know which one of those were. But anyway, the last report, they have no alewife seed in the lake and many water fleas and clear lake. Pretty much a complete success story. Next slide. So these were large lakes. What about smaller lakes? Smaller lakes are more intense, allow more intense management because it's more affordable. And one management is the phosphorus sequestration. Chemicals in the lake, you put chemicals in the lake to strip the phosphorus from the water column and lock it into the sediments. Or aeration of the hypolimnium, that lower level, can prevent internal loading. Weed treatments like herbicides and harvesting. And also some lakes just put blue dye in their lakes, and that attenuates the light coming in and the algal populations decrease. Next slide. Oh, can you go back to that one? Okay, so what are the take-home messages? And this is all caps. Prevention is always better than mitigation, and it's a lot cheaper. Focus on phosphorus. Lakeshore lawns are an important part of the equation, especially for lakes with good water quality. If a lake has lousy water quality, the lawn, working on the lawns isn't going to make a big difference. Lake associations are on the front line for lake protection. They must keep aware of their nutrient levels, and CSLAP, which is a statewide way of measuring, a place where you can measure your water chemicals. And they write a report and give you some warnings. So look at those warnings so you know how that the lake is changing, and the earlier you get to it, the better. Next slide. Okay, this is a quote from a Senegalese environmentalist. In the end, we will conserve only what we love, and we will love only what we understand, and we understand only what we are taught. And with that, I'll ask for questions. Okay.
All right, thank you, Peter. Much appreciated. That was fascinating. I hope everyone enjoyed it. We do have a couple of questions in the chat. Here's the first one from Everett. Everett writes, on Scroon, we see fish on our depth finder at 60 to 100 feet in the summer. Does that indicate adequate oxygen levels? And then he's also asking, what oxygen levels do lake trout and landlocked salmon need?
Better lake levels than what we have in Sacandaga Lake. You know, 4 to 5 to 6 parts per million oxygen, something like that. When you get down around 2 to 3, those fish will go up into the thermocline where it's a little bit more oxygen. It's very interesting because I know that the oxygen levels in our lakes are pretty low in the summer, but there's a fishing guide down in Wells who catches big fish, and he says he's catching them down below. So I don't know what the heck he's doing. So usually you would think that the fish would not be in the hypolimnion when the oxygen levels are low, but he says he can catch fish then. So I don't know the exact levels, but it would be somewhere in the 4 to 5, and the fish, of course, can migrate up and down in the lake. But I don't know the answer to that precisely, but 4 to 5 to 6 to 7 somewhere would, and it would depend on the fish too, of course, and the conditions that the fish are in. But I hesitate to give a number that because I'm not positive. I
Right,
don't know whether that helps or not.
Paul asks, does Sacandaga have a constant flow through inlet to outlet?
Yes, we have about a 2-year turnover rate in our lakes, so which is very, very nice. So yes, I do, and the inlet is a bunch of tributaries, and the outlet goes into Lake Pleasant, which is the origin of the main branch of the Sacandaga River.
And we have a follow-up. If so, it sounds like it is so. How does that affect your measurements and the thermocline data?
How does what the flow through? Is that what?
Yeah.
That have nothing to do with that at all. That's a much slower process, and now. Well, it gets complicated, but it does as far as the stratification of the lake, which is the formation of the formation of the thermocline, it won't have any effect at all. But as far as where pollutants are, if a pollutant is coming in the summer from a tributary, that would be relatively warm water, and it would stay in the upper layer. It wouldn't even go down into the lower level of the lake. Because the source would be warm water. So I don't know whether that helps, that answers his question or not. But the rate of exchange doesn't have much to do with the dynamics of the upper and lower levels of the lake.
Bill asks a good question here. The application of salt to roads in the winter increases lake salt concentration. What are the effects of that?
Of course, it depends on the level. Well, at lower levels, there's not much of an effect, but at really high levels, which some of the especially urban lakes have, it can have a big effect. I think that the person who you should know to talk to is a Hetling over there and up in Paul Smith's. He's done a lot of work with road salt. It doesn't seem to have at lower levels, which most lakes have. It doesn't seem to have a huge effect yet, but of course it's accumulative. Although with with your lake turnover, with the flushing rates of the lake, it can flush some of that stuff out. Anyway, so it's not going to be totally accumulating. So that doesn't help much. It doesn't seem to be affecting Adirondack lakes much yet, but in the future it might. So I'm not sure about that. That's not much help, but anyway, because quite frankly, I'm not sure we really know.
Peter, I'll chime in on that. One of the lakes that we've done, that researchers, especially at the Adirondack Watershed Institute at Paul Smith's College and the Ausable River Association have done, is looking at Mirror Lake, which is probably our most developed lake in the Adirondacks, and looking at the salt concentrations. It had some of the highest salt concentrations. And one of the things that they were starting to see with that was that the salt concentrations are doing a very similar density effect. The salt water is changing the density, and then that density is influencing the natural turnover of the lakes because it's making it harder. The density of the salt can make it much harder for it to move through. So that was one of the things that spurred Lake Placid and the surrounding communities to try to start controlling it. They started to bring down the levels of it. So yeah, we're starting to see some impacts, and like Peter says, the salt and some of these lakes can be cumulative. It just can kind of add up over time.
And we'll do one more here, and then we'll move on to Brian. Kathy asks, We are moving forward with a plan to use Priscillicor to reduce the Eurasian water milfoil in Chateauguay Lake. Will the die-off from this pesticide cause a substantial increase in phosphorus levels that we need to be concerned about for possible algae blooms afterward?
It will be interesting to see. I don't know the answer to that, and how quickly it will be. It will be. It will be chopped up or disposed of by the organisms down there. It will depend on whether the lake is stratified or not. Where in that case, it would decrease the oxygen level significantly in the hypolimnion. Now, as far as phosphorus goes, I'm sure that it would be there would be more in the lake. Whether it would be measurable or not, I don't know. But,
yeah, I'll chime in on this one a little bit, Peter. So Eurasian water milfoil has this annual plant growth. So every year it's growing as a submerged plant from the sediment up into the water column, and then it dies back in the fall usually, and then regrows from the root. So Eurasian water milfoil and other plants, they are actively taking nutrients out of the sediment as they're growing with their roots, and then when they senesce or die, the portion in the water that re-releases those nutrients back into the water column. So one of the things when we do chemical management of Eurasian water milfoil is we generally are targeting that very early in the season. So that's usually late May, early June. So we are treating it before there's a lot of biomass. So when you're doing it that way, there's not going to be as much nutrients going back because the plants have not been able to. It's not like the full plant growth at the end of August and September. That happens every year, where they're just naturally dying and naturally putting those nutrients back into the water column. So when you are doing that treatment early on, there's less plant material that could contribute to dissolved oxygen issues or also nutrient issues. So unlikely, but APIPP is working with some partners where we are looking to do some water quality sampling to help us better understand this.
Good.
All right, and there are a couple more questions in the chat, but I think we should save those to the end. We should get moving along here. Brian, if you want to take control of my screen, we can get into lake ecology and invasive species. Okay, I got it. There you go.
You're muted, Brian.
Okay, also let me get back to this now. Sorry about that. Sean, if you bring it back up to where I am,
Yeah.
then we can get me started.
No problem. Apologize for the technical difficulties here. There we go.
All right, so I will request remote control.
Okay.
Here you go, through the magic of the satellites and the internet. So Peter did a great job of talking about all these different lake ecology and different processes that are going on with the lakes. And I'm going to talk about how invasive species can influence some of those. And I'm going to give you 2 examples. So like Peter said, most of the time when people come up and they visit our Adirondack lakes, they're looking out at the water and they're seeing these beautiful scenes, the mountains, the forest, and they think of these as very peaceful and tranquil, kind of places that are kind of static in their minds. But as Peter talked about, they're really active and changing and evolving places. And so, our lakes are really dynamic. And Peter talked a lot about the seasonal change, the turnover, the different densities with the different water that's going on them. And then he also talked about the food webs and how, there's so many different interactions and things happening between the phytoplankton and the zooplankton and the fish and the crustaceans and the birds and the people up in there. So, this is an example that I got from the Hudson River. And the thing about aquatic invasive species is that they really are an unplanned experiment, that when we move a species in there, we don't know all the impacts that they likely can have. And many times it's not until years later that we're able to see what is going on. So, when you add in a species, like in this food web example, the zebra mussels, they can have some pretty large impacts on these different systems, and some of them can be what we call ecosystem engineers. So the first one I'm going to talk about is zebra mussel. This is a mussel that is native to the Caspian region. So think like the Black, Caspian, and Aral Sea of the Eastern Europe. It was brought to the Great Lakes in the 1980s via ballast water of cargo container ships, and has since spread all throughout the Great Lakes and the connected waterways, and spreading throughout the eastern United States, also into the western United States now. It was first discovered in Lake Champlain in 1993, and it has this kind of zebra characteristic striping that you can see here. The striping can kind of vary depending on the size and the age of it. Most of these mussels are very small, less than an inch or half an inch, as you can see with the picture of the penny right here. And these mussels are filter feeders. They filter the water, and they are eating things like phytoplankton, things like in the water, like bacterias and viruses. They're absorbing organic matter, debris, and then some of the zooplanktons, the smaller zooplanktons, they can actually eat themselves. And one individual zebra mussel can filter up to 1 liter of water every day. So, so if you think about it, in that lake, we have, those pictures that Peter was showing. There's all sorts of zooplankton and organic matter and bacteria and all sorts of things that are moving around in the lakes, and these zebra mussels are filtering that in and out of their bodies. And an interesting thing that they kind of do is that, they're reducing the overall amounts of these in the water bodies, but they can also be selective about them. There's some new research that shows that they can selectively choose what they want to take in and keep and what they want to reject. So there's some data that's showing that they can sometimes preferentially reject more of the cyanobacteria that are responsible for the harmful algal blooms. And so this is just another example of how they are changing the competitive balance. So they are removing the zooplankton and the phytoplankton and the good things that we like, and then putting more of the bacteria out there that are causing problems. Another example that we've had from the Great Lakes is where they have been doing some bioaccumulating, some botulism, a form of avian botulism. And then when another invasive species around gobies eats the zebras, they are upcycling that through the food chain. And it has been leading to large die-offs where thousands of birds, things like loons and cormorants and other birds, grebes, have been eating them and then dying themselves. So, and you might think like, okay, like 1 liter of water. So here's these photos from Lake Simcoe Conservation Authority, where they took some water out from the lake and they put some zebra mussels in one of them. And then after 10 minutes, you can see that the photo on the right is much clearer because they've filtered out that water. And well, in the little these little containers, it doesn't look too much. But when you really start to multiply that out, these zebra mussels can grow in incredibly high densities. Hundreds of thousands of zebra mussels, per square meter. Here's a picture from Lake Champlain where I found these zebra mussels growing on the shells of native mussels. And so they grew so much on the native mussels, I believe this is an Eastern Elliptio, that they prevent it from being able to move. The native mussel can no longer move, and so they're killing them. But you can just see like how much they will cover. They'll cover every single thing. They cover your docks, your boats, pipes. There's a lot of famous photos of people dropping things in like shopping carts or tires, and you pull them up, at the end of the season, and they're just completely encrusted. So when you get that many of them, they can have a really large ecosystem level impact. And one of the things we're able to see that is in the water clarity. So this is Lake Simcoe, once again, in Canada, where they have been measuring the water clarity with the SESI dish. And you can see that pre-invasion in 1995, the average depth was about 3.8 meters for the water clarity in it. Well, after the zebra mussels came in, there was this sharp drop off, and they almost doubled the water clarity, and because they've removed out so much of the algae and the phytoplankton that's in there, and so they increased it to over 7 meters. And so then when you look at that, it really took that lake from being a algae dominated turbid lake where phosphorus was kind of distributed throughout the water, the column, both open and the littoral zone, to once it came in, it cleared up the lake. That made the water clarity, the light, be able to reach much deeper. Now the plants could grow deeper down in the lake, and so it moved to a plant-dominated lake on that. It pushed the thermocline down deeper in some of these lakes, and so the fish have to go down lower. These zebra mussels are also filtering stuff in, and they're excreting it out. So they're now putting more of the phosphorus in the near shore environment, where it's leading to algal blooms and some other interactions going on. So this is an example how of a small mussel, when it gets to such large level, it can start to change some of these ecological processes that we see, and really start to change like the trophic states and the thermoclines and other these processes that Peter was talking about. So, you might say like, oh, okay, well, that's an animal. Like, we're always used to hearing stories about animals changing their environment or changing our ecosystems. Well, what about plants? Well, we're going to talk about Eurasian water milfoil. This is a plant that was originally native to Eurasia and North Africa. It was introduced to North America over 100 years ago, and it is our most widespread aquatic invasive species in New York and in the Adirondacks. It's in around like 68 some different lakes in the Adirondacks. So it's one of the most widespread invasive species in the United States. And you'll say like, well, it's a plant. Like, we have lots of plants that grow in our lakes. We even have native milfoils in the Adirondacks for tens of thousands of years. We probably have had native species of milfoils. What's so different about this one? It can grow it's a submerged plant. It can grow at the depths up to 15 or 18 feet in really clear water. And you'll see it start to come to these places and, go from like a low density to kind of like a medium density that you see in this photo here. And then it can grow up into, if the conditions are right in some littoral zones, it can just become a solid monoculture, just incredibly dense bed. This picture on the right, it formed this complete mat of where this lake I was working in, Lake Alice, and you can start to see all the little flowers of it coming up here. And once these plants start to get to these really large levels, they can have large impacts on our lakes. And so, yeah, here's the picture on the left. You can see it, a solid carpet, a solid, mat of it on top of the water. And so think about our lakes, as Peter was talking about, we have our thermoclines, we have where the places where light can penetrate to, where the fish, want to live. And so that sunlight is penetrating through the water, allowing the plants to grow, to do photosynthesis and produce oxygen in the lake. We're also having that diffusion that's happening so that the atmosphere has a lot of oxygen in it. It's diffusing into the lake through the lake surface, and then things like the wind and the waves and the entrainment that helps put dissolved oxygen. So that increases, our dissolved oxygen in the lake, which is important for the organisms living there. So what happens when you get a solid mat of Eurasian water milfoil? And as you can see in that picture on the left, like when it grows up to the surface, it doesn't stop growing. It just starts growing horizontally then. So it can be incredibly thick in some areas. Well, what's first going to happen? Well, now the sunlight isn't able to go through that dense mat. So the sunlight is just heating up that top part of it. So when I put my hand in this water, that upper 6 inches felt like hot bathtub water. It was unreal how warm it was. And then if I kept pushing my hand further down, it got colder once I broke through that layer. Once again, that's doing that thermal stratification. Now the water in the really warm layer is not mixing with the cold layer below it. That layer is also blocking the sunlight from going down below to it. And it's also blocking that really gas exchange, because now we just have this really warm, stagnant water up at top. There's no places for the ripples and waves to entrain it. So that's going to make the dissolved oxygen levels start to go down. And so all of that is going to, impact the organisms that live there. When they form these really solid mats, there's no sunlight that's able to go through. The native plants can't live underneath there. The dissolved oxygen levels start to go down. So the macroinvertebrates, the zooplankton, the fish, they can really start to suffer from that. And, then because we are having this just like stagnant, really warm water in there, just to add insult to injury, we are create a perfect breeding ground for things like mosquitoes and maybe some of the other insects that we don't really like. And you can see these really big algal growths kind of starting to occur on there. So, this is a good example of how a plant can come in and it can start to change the water quality. So, overall, those are just 2 examples. There's many more where aquatic invasive species can have impacts on these lake ecology. Things like the food webs, the water quality, the temperature, the clarity of it, the biodiversity, where they're either directly killing, eating, or out competing the other native plants and animals. We can have some examples where the invasive species can grow, especially the plants can grow so dense that they prevent the movement of water and cause flooding. And, all of these things are what we call ecosystem services, the goods and benefits that we get from our lakes, and they can start to impact the things that, people really care about, like recreation or public health or the economy. So, it's really important to remember that these aquatic invasive species are this really large, unplanned experiment and can have these sometimes very large impacts on our lakes and their ecology. So, well, what can we do? Well, when we talk about this, I kind of break it down in these 3 bins. We try to prevent, we try to monitor, and then we try to manage invasive species. And this can have us have healthy lakes. So one of the things that you can do, you can join our lake protectors. We have many lake protectors I'm seeing here on the Zoom, and we train people how to identify 16 plants and animal species that we are tracking across the Adirondacks, and they report it through the IMAP invasive systems. So we've had hundreds of volunteers from just the general public to different partners like schools and communities and nonprofit environmental groups do this. So we'd love to have you come and join us this summer and choose a lake that you're really interested in, and you go out and you report what you see or what you don't see for aquatic invasive species. The big part that we often talk about mentioned is prevention. It's much better to prevent an invasive species than to try to have to manage or deal with impacts of it. So the most common pathways of spread through water, canoe, fishing, when people fishing, water body. Sometimes people aquarium pets. I'm going to do a good thing and save them and go to the local stream or lake and just dump them into there. But actually, that can cause a lot of problems because those species can become invasives themselves. A big part of our prevention program here in the Adirondacks is the Adirondack Watershed Institute and their boat stewards that they have stationed all across the Adirondacks. And that's really important because we get visitors from over 40 different states, 6 different Canadian provinces, up to almost 100,000 boaters a year. And they're coming from these places that have a lot of invasive species. Things like the Great Lakes, Lake Champlain, the St. Lawrence River, the Connecticut River, the Hudson River. And so, we are at risk of being invaded from them. And so that's why these boat stewards are a great protection, and they can help you follow the steps of clean, drain, dry, which is the most important thing that we can do. Any of your boats, water skis, fishing gear, anything that touches the water, it needs to be cleaned. Remove anything that you see, drain all the water out, and then let it completely dry to do that. And one of the best ways to do that is to visit a decontamination station. And so these decontamination stations, they have these high-powered pressure washers that have hot water that can kill some of those really small life stages of the zebra mussel or the spiny water flea. And you can go get a free boat wash and get cleaned off, and then that greatly reduces the likelihood that you're going to transport aquatic invasive species from one lake to another lake. And it's not only the right thing to do, it's actually the law in New York. We have a law that requires everyone to follow clean, drain, dry for all their boats and equipment. And in the Adirondacks, in the 10-mile zone around it, all motorboats are required to have a certificate stating that they have followed clean, drain, dry. So I think Sean will go next.
Yeah, thanks,
wrap
Brian.
us up.
Yeah, thank you, Brian. So we're just about at 11:30. I want to give a quick shout out for some upcoming events, and then we will take questions. So I urge everyone to please stick around. There are a few questions in the chat. April 25th, we have the APIPP Spring Partner Meeting. In May, we have a webinar, Invasive Species at Our Door. So this is going to deal with invasive species that are not quite here in the Adirondacks yet, but we're keeping an eye on them. We don't want them here. I think that's going to be a really interesting discussion. And we haven't set a date for that one quite yet. We're waiting on confirmation from one more speaker, but keep your eye on our website. And then on June 5th, I'm really excited about this one. The June 5th is part of New York Invasive Species Awareness Week. And for that week, we are going to do a trivia night at Racket River Brewing at Tupper Lake. There's going to be trivia. There's going to be a special beer that they're making. It's an IPA that we're calling Invasive Plant Ale. Yeah, and we'll be there all day, just talking to people. Then in the evening, we'll do the trivia night. So that's gonna be really exciting. We hope to see you there. And to learn more or register for an event, as always, visit adkinvasives.com/events. And our recordings are available on our website, or you can just head over to our YouTube channel. And this one will be up in about 24 hours or less, I would say. And lastly, APIPP has a bunch of free outreach materials. You can order them from our website as well. You can also download them from our website. We have Protect Your Forests and Protect Your Waters brochures and posters. They give some really helpful tips and reminders on how to help prevent spreading invasive species. And then we have the Protect Adirondack Trees, Buy It Where You Burn It rack cards. And those are just a reminder to not move firewood, buy it where you plan on burning it. Don't bring it because you could be carrying invasive insects and pests along with you. And lastly, we have our terrestrial invasive species of the Adirondacks field guide. This is also free. It covers about 28 invasive species in the Adirondacks, and it really does work as a field guide. It teaches you how to identify them and also how to report any invasive species that you might come across in your adventures. So now we'll field some questions. Let's see. We'll jump right into, and Brian or Peter, you can feel free to answer these. Do you have an opinion on the macrophyte inflated bladderwort? Some states list it as an invasive species, but New York does not. It is now found in several Adirondack lakes.
Yes, so we have many different species of bladderworts. They're called the genus Utricularia. There is one species, inflated bladderwort, that is native to we're fairly certain it's native to the state of New York. We have herbarium records for it for a fair amount of time, going back in history in the more southern region of New York. The question is it native to the Adirondacks? And that's a question that we haven't conclusively settled yet. So there are some lakes like Len and Racket Lake where they're starting to see really large growth of it, where it's starting to become kind of problematic. So yeah, that's definitely a species we are keeping a radar on and trying to work with regional people to kind of learn more about.
And this one's actually from a little earlier. Want to make sure we don't miss it. With a high flow lake, does that affect the amount of seasonal turnover?
Seasonal turnover, meaning the flushing rate of the lake. The more water you put in there, the quicker it's going to go out because the volume of the lake stays the same. So the flow through would be greater if you had a bigger flow into it. Was that the question?
I believe so, yes. If the person who asked that could come off mute if he didn't answer the question and clarify, but that's my understanding, yes.
I think one of the things they're also kind of thinking on, and you can just kind of think about this, like logically, part of the things that we're talking about is this thermal stratification of lakes, and to do that, the water needs to be, have a slower residence time. Like in rivers, we don't see a thermal stratification of most rivers because the water is moving so quickly. It's not there to kind of form these different layers in them. And so, in the lake, in most of our lakes where the water is staying longer, that is a seasonal pattern, that happens every year.
All right, another question from Bill. What steps can be taken to fight zebra mussel infestations?
Well, the first step is keep them out, because once they get in there, they are really hard to deal with. There are almost hardly no real examples of once zebra mussels go in a lake that you can eradicate them. So once they're there and at a very established populations, you're really just trying to suppress them kind of locally. They can become problems at with human structures on docks, on boats, water intake pipes, and different things. So there's lots of different products like chemical coatings or paints for your boats. Mechanically having to go and clean these these structures to remove them off. So there's some localized things that you can do, but they are generally stopgap measures. You're just kind of removing the buildup that's there, and then they'll slowly repopulate, and so you just constantly have to be doing that.
Hey, Brad is wondering, is there Adirondack evidence of fish communities shifting with Eurasian water milfoil invasion?
I don't know.
Yeah, I don't know if I've seen like a good documented example from the Adirondacks. We do have some different examples of from like kind of the Midwest, Wisconsin, Minnesota, showing some impacts of changing plant communities on the fish communities. also some changes of fish communities with invasive mussels and invasive water fleas, like spiny water flea or zooplankton in there. Initially when to some of these, fish and, the Eurasian water milk is just another plant. And we've had native milfoils, so, they're used to living it. It's the problem is that when they get too dense. So, fish will use Eurasian water milfoil as habitat and, there are insects and things, growing off of them. But the problem is that when it gets so dense that it becomes these monocultures and we lose that kind of diversity of different plant habitats that really can start to impact. And then, like when they become that solid mat, like that lake Alice example I gave, like it can be really hard for the fish to survive, under some of those conditions.
All right, and Leonard asks, who is responsible for responding to a new introduction of an invasive species to a water body? Is it the state or county?
And Sean, I'm working on a long answer to Leonard, but we can have our presenters
Gotcha.
answer as well.
Okay, thank you, Tara.
Yeah, so it is that's a little bit of who's responsible for it, as in who is who's the person that's going to manage it and help do it. I mean, who's responsible for it is ultimately most times the human, whoever brought it in, but we generally don't know who that human is. So it's all of us, and so it's on all of us to follow those clean, drain, dry procedures. And then who are the groups that do the management or try to deal with those impacts? Well, that depends on a lot of different factors, like who owns the water body, what's the communities that are around it. But in the Adirondacks, the vast majority of management efforts are done on the local level. It's done by the local lake association, the local town, communities. We in the Adirondacks, we spend over $1 million a year trying to manage and remove aquatic invasive species, mainly Eurasian water milfoil, and most of that is paid for, through a variety of different things. Sometimes it's some state funding, sometimes it's local funding, but a lot of it is private funding from these lake associations.
Brian, everybody should realize that the lake associations have a major responsibility here, in my view. And I think the sooner you can find these things, the better. And one really good thing is to, what we do in our lake association is that we've educated lakeshore owners what these things look like, and they survey their shore once or twice a year, and if they find something weird, they give it to me and or another member of the committee that knows about them. And so that every year we have essentially a survey of the lake by our educated lake shore owners who are members of our lake association. The lake associations have really got to get their acts together with regard to these things.
Yeah,
Brian, go ahead, sorry.
Yeah, our local communities really are the stewards of our lakes, and so we depend on them to be active and to be engaged.
Brian, I think this one's for you. Can you say something about the value of NYSFOLA and CSLAP membership?
Yes, I am a strong supporter of NYSFOLA and the CSLAP. I hope people can come out and see me at the NYSFOLA annual conference in Lake George. I'm going to be talking about our lake management tracker program. So NYSFOLA, the New York State Federation of Lake Associations, and they help implement our CSLAP, Citizen Science Lake Assessment Program. Did I get that right, Peter?
Yeah, but the CSLAP, yeah, Citizens. So I think a Citizen Statewide Lake Association.
Citizen Statewide Lake Assessment Program. We also have another one called ALAP, the Adirondack Lake Alliance Program. And excuse me, ALAP, Adirondack Lake Assessment Program. And then we also have the Adirondack Lakes Alliance. So we have a lot of different coordinations and different groups, but the main point is, we can learn and share from each other. And the CSLAP and ALAP program the real key about them is that it's doing that long-term water quality monitoring. So, how Peter was able to talk about and, show those data about, what is your phosphorus levels and, parts per billion, and, what is your Secchi depth and the water clarity. The only way we know that is if people go out and monitor, and vast majority, a big part of that monitoring is happened by, volunteers and trained individuals from environmental groups. And so, yeah, every lake should be participating in either CSLAP or ALAP every year to monitor the water quality.
I second that, and this is one way that you can get long-term data on your lakes if you are involved with the CSLAP data. And for instance, in the Sacandaga Lake, we've been in the CSLAP program since the mid-'80s, so we've got 30-some years of data on that lake, and it's really quite valuable. And really, that's the way you can keep tabs on your lake and find out what's going on with it.
Yeah, so just like we want people to be monitoring every year for aquatic invasive species, we also want them to be monitoring their water quality.
You don't have to monitor the water quality as frequently as you do the weeds. You got to do the weeds every year, if more, not more. and you can have alternate years or every third year on lakes. Anyway, so spend your efforts on the weeds, but also get the other ones done too.
Great, thanks. Is there a pamphlet that gives examples of native Adirondack plants to use for a buffer between lawns and water body? I know APIPP does not have one, but do Brian or Peter, do you know of one?
There's a book that's out called Lakescaping, and you can probably buy that on Amazon and things like that. A valuable partner in this is your soil and water conservation people. They would have really good ideas, and they actually sell plants that you could use for your lake buffer. So I would check with them.
Yeah, there's several programs, that different groups run, like Lake-Wise or Stream-Wise, the Lake Champlain Basin Program, the Adirondack Watershed Institute. Well, they can have people who actually come out to your property, and they can do, look at what your riparian buffer is and your lakeshore buffer, and kind of can suggest plants or things to grow.
So we have a few more here. Thanks everyone for sticking around. For Peter. Oh, sure.
Sean, can I jump in quick with one more thing?
Yes.
Thanks, Sean. I just wanted to add our partners over at the AuSable River Association also did a great project 2 years ago that they presented on last year, where they contracted with Steve Langdon, and they really drilled down into native plants and grasses to be used in riparian areas. And they have a great webinar about that. I'd be happy to drop that into the chat as well.
Thanks, Becca. A question for Peter. What do you think the top 5 threats are to our Adirondack lakes?
Phosphorus in your lake, invasives, animals and plants, lawn buffers. Is that 5? I don't know.
That's probably close enough.
Invasive species. Anyway, those are the main ones. Everybody should be aware of what their phosphorus levels are in their lakes and how it can be managed.
Yeah, and I'll add in climate change, and that kind of gets us into the upcoming thing is that when you talk to lake limnologists and ecologists, one of the biggest changes that we are having up here in the Adirondacks is we're having a lot more energy going into these lakes, and the amount of time when ice is on our lakes is shrinking, and we're pushing apart the distance of turnover. Our summers are lasting much longer now, and so these are impacting some of these lake processes that we've been talking about.
Yeah, the ice-free times is really critical. It's certainly critical with regard to the depletion of oxygen in the hypolimnion. The longer time that between turnovers, the higher the chances are you're going to get low oxygen levels in the hypolimnion, as well as plant growth. Plants have another month to grow than they used to have. So there are big things that are going on with regard to climate.
Wow. Can one of you, Peter or Brian, talk a little bit about the value of genetic testing on macrophytes?
I can chime in on that. So we are been working with some different partners, and we're starting to do some projects. That's from some research institutions, trying to look at the genetic diversity of plants. A group of plants we're specifically interested in is the milfoils, because we have both native milfoils and invasive milfoils, and these invasive milfoils can actually start to hybridize with some of the native milfoils. And there's some research from different parts of the country that show these hybrids can have what we call hybrid vigor, that they might actually have some competitive traits that can outcompete both the individual natives and the individual invasive species. They might be more resistant to some types of management. So yeah, this summer we're partnering with SUNY Oneonta, and they have a Lake Champlain Basin program funded research project that's going to look at the genetic diversity of milfoils all throughout the Lake Champlain basin. So yeah, that's one example of it. And if you think about each of these lakes, they are kind of their own little systems in the environments, and so there's a lot of genetic diversity in there, and that's one of the types of diversity we're trying to protect.
That is all I'm seeing in the chat. Becca did put a link on native plants and grasses for riparian areas in the chat. Please head over there and check that out if you're interested. I may have missed a question on accident. Does anyone have any other questions before we end the webinar? And feel free to come off mute or type it in the chat if you do.
Could I throw something out here real quick?
Yes.
Earlier, I believe someone from Chateauguay asked about algae blooms following Priscillicor use. I'm with the Warren County Soil and Water Conservation District. We've had 2 lakes here in our county use Priscillicor in the last 2 years. Sunnyside Lake, small kettle lake down in the town of Queensbury, mostly sandy soils. No inlet, no outlet. They used it 2 years ago, and in year 2 after treatment, we did see pretty substantial algae bloom. It may not be the only causation there. We also have tremendous number of homes around it, all of them on septic systems. And last year we had an extraordinarily rainy July and August. So that may have also been a function. And because it's only a 20-foot kettle lake, no inlet, no outlet, it warms fairly quickly in the year, and they had substantial biomass formed by the time they used it. So it killed every piece of milfoil we could see. We found no living plants after 60 days, and almost no living plants actually after 30 days. And we did have that biomass drop to the bottom and break down over the year. So we can't make it a direct correlation. But we also had the Lake Luzerne used it last year. So this summer when we go out and film the plant community, we'll be taking a look to see if there's a noticeable algae bloom in the first season after treatment this year for them. But it's a much deeper lake, has a much quicker turnover, obviously, since there's probably little turnover in a kettle lake. So we'll be able to make more statements about that after we take a look at 1 year plus 30 and 60 days in Lake Luzerne.
When you get rid of the plants, the phosphorus has got the phosphorus input to the lake from external sources and things is going to be constant. So there's going to be more phosphorus available for algae. So that may be the reason for the blooms as opposed to the decomposition because of Procelcor.
Yeah, that's why we want to take a look at in Sunnyside at maybe expansion of natives. It's nearly, there's probably 90% littoral zone in this kettle lake. So there's only a very small area that doesn't hold plants. It has a robust native population. So we're more interested in looking at year 2 and 3 after treatment to see if it continues to have some algae problems there. And we'll be also interested in seeing what occurs in Lake Luzerne. That's it for me.
All right. Thanks, Robert. Does anyone else have any questions? All right, we have a lot of thank yous in the chat. You are welcome. And if there's no more questions, I'll send out a follow-up email with a link to this webinar. Please share it with your friends. Have a good day, everybody.
Good. Brian, are you still there? No, I guess not.
I'm still here, Peter.
There was data on zebra mussels in the lower Hudson that